Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO, and your host. Today, Dr. Steve Ramirez will present Manipulating Memories to Help Treat Brain Illness. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 183 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such a great impact is because we don't limit our focus to one illness or condition. Since 1987, the Foundation has awarded more than $430 million to fund more than 6,200 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Steve Ramirez. Dr. Ramirez is Assistant Professor of Psychological and Brain Sciences at Boston University and was a 2016 Young Investigator. Our webinar will begin with Dr. Ramirez's presentation, which will then be followed by questions and answers. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Ramirez. Steve, the floor is yours.
Thank you so much for the incredibly kind introduction, and it's such a pleasure to be here today. I'm really excited to go over a lot of the work that has really accumulated in the last decade, in particular of memory research, and then to kind of pivot into what my lab is doing these days. But I really appreciate both the opportunity to be here, and looks like we have a pretty great crowd too, looking at the attendees. So thank you everyone for attending the seminar today. I'll start off with this quote from a fortune cookie that I thought was pretty powerful, which was that if the brain were so simple we could understand it, we would be so simple we couldn't. And I think that really speaks to this kind of Mobius strip of problems that we have in neuroscience, that we're using the brain to try to understand itself. And I think that's powerful because that really, I mean, it speaks to what human endeavors are truly capable of. And I really want to chart out the remarkable progress that's happened in terms of the brain trying to understand how it remembers. Like, how does the brain store information that it experiences about the world, and how does it retrieve that information that it experiences of the world to guide how we conduct ourselves in the future as well. So the structure of the talk today will be, we'll start off by going over a bit of work that I did as a grad student, because it really sets the stage for what my lab currently does. And then from that, we'll go into some more recent unpublished work from the lab that I'm really excited to share with everyone, as well as just to think really big picture. What does this all mean? Like, what does it mean to try to reverse engineer the time machine that is memory in our brains? And ideally, can we reverse engineer it in a manner that pinpoints to treatments, like to how to fix the car, so to speak, when it breaks down, and to really view memory. What I'll argue here towards the end is that we can view memory itself as a kind of treatment for the brain. And then in this case, well, I'll end again with big picture applications of what we're doing in the lab to try to really give memory a kind of medicinal power that maybe went unappreciated beforehand. So I'll start off here with just asking everyone in the audience, like, what does what does memory mean to you? This is just a copy paste picture from the movie Inside Out. And I'm curious what everyone is thinking about in terms of what memory means to you, because my answer has really changed over time. Like when I was a college student, memory used to mean, it was kind of boring. It meant memorizing textbooks. It meant memorizing facts to regurgitate on an exam. And it wasn't really until years later that I really began appreciating the kind of mental time machine that memory truly is. Like I make fun of my physicist friends saying that in between all of our ears, you have the one thing that physics tells us we can't actually have, which is a time machine to the past. So all you have to do is just close your eyes and think about something as seemingly straightforward as what you had for breakfast today or what you did yesterday. And that's it. In the blink of an eye, you've time traveled back to a moment of your past without breaking a sweat. It's impressive that a brain can do that. Like it's impressive that your entire self can be transported back to any moment of your past and to really let it almost absorb you. I mean, I'll talk about this a little bit towards the tail end of today. This idea that when you're absorbed by a memory, when it's good, it can feel great. And when it's bad, it can be pretty debilitating. So memory has this kind of bidirectional power to really put us in an unbelievably positive headspace, or to put us in some darker corners of what past experiences have really left in terms of a mark on our brain. And I think both are important. And one of the things that I'll really lean into throughout the talk is that memory is, it's even more powerful, I think, than I ever appreciated as an undergraduate or as a graduate student. So to start off here, one of the first questions that we had with regards to memory was, where is a memory located in the brain, right? Seemingly straightforward question. We know that somewhere in between your ears, memories exist, but where are they located? And then one of the first questions that we really had was, if we could find a memory, could we try to artificially jumpstart one? Could we try to hotwire the car, so to speak, to try to jumpstart this process of recollection? We wanted to do this to see if, one, what does this tell us about the brain? But then again, can we actually use memory for some kind of beneficial purposes? So we're really standing on the shoulders of giants when we're starting off these first experiments, because the idea of going in and finding a memory in the brain can be pretty daunting. You have 86 billion brain cells and 100 trillion connections between those brain cells. It's a pretty complicated map to navigate. So in the 1940s and 50s, the Canadian neurosurgeon Wilder Penfield would go and perform surgery on a handful of patients with epilepsy. And what he would do was go and stimulate certain parts of the brain and ask, does it look like a seizure would actually be onsetting or not? And now the amazing part of this, and I think it's one of the best footnotes of neuroscience here, or one of the most profound footnotes in science, was that when Penfield went and stimulated in an area of the brain that's right above your ears, tucked inwardly, the temporal lobe, a subset of patients reported, like, doctor, I don't know why, but when you stimulate there, I'm randomly remembering my prom when I was 16, and I can hear the music faintly in the background. Or other patients would report, I feel like I was just transported back to when I was walking my dog with my grandma, and I can vaguely make out the trees and vaguely make out the street that we were on. And there were pretty vivid remembrances. And I thought that this was pretty amazing, because this was 70 years ago. And yet going in and stimulating a part of the brain, the temporal lobe here, was enough to kind of elicit what I guess contemporary Hollywood would call total recall, right? It's like being able to go in and stimulate a patch of brain, and that was enough to jumpstart this remarkable process of recollection. That gave us hints as to where we could actually go in and start to see if we could identify a memory, let alone control a memory. So we zoomed in when starting these experiments into the temporal lobe, and underneath this flap of temporal lobe, there's an area of the brain that I think most people are acquainted with, called the hippocampus, which is an area that we know is important for learning and memory, and its deterioration leads to pretty profound amnesia. So hippocampus is one of these really important areas of the brain that we know are involved in both processing and recalling our daily experiences, right? The things that we hold near and dear. So the first goal here was, can we look at a memory? Can we peer into the brain and see what one looks like? We use rodents as mammalian subjects. The hippocampus is an area that's pretty nicely evolutionarily conserved. It gives us, and the rodent models here give us some pretty powerful genetic strategies to go in and try to identify memory-bearing cells, like cells that we think hold on to an individual memory. So what we ended up starting off with was we were both iterating on previous systems and developing new systems for finding the brain cells that hold on to an individual memory. And could we actually go and literally look at them under the microscope to get a kind of sense of what a physical cross-section of a memory, if you will, looks like? So we developed a handful of genetic strategies to find cells that hold on to an individual memory. And first, we just wanted to see what it literally looks like. And now these are two images here. This is a cross section of the hippocampus that I mentioned. And now the cells in green, the green dots here, are cells in the rodent hippocampus that we believe were holding on to a negative memory. And then below here, the cells that are in this kind of avatar cyan blue color are the ones that we think were holding on to a positive memory. But this is one of my favorite images I've ever taken as a scientist, because it really spoke to me in the sense that, you know, memory is something that doesn't really feel like you should be able to touch it, right? Like memory is this ephemeral, subjective thing that we all experience and we all have. It doesn't feel as something that you could physically put under a microscope and actually go in and poke with a small enough microscopic finger, and yet here we are. So this was what I consider the crystallization of a memory in the brain, but it gave us a proof of principle that we could go in and actually find the brain cells that hold on to individual experiences and look at them. And then we wanted to one up that and ask, given that we can see which brain cells are involved in a memory, can we turn them on? Can we activate them to see if that would jumpstart this process of recollection? So I'll go through one experiment here, and it really sets the tone for what the rest of the talk will be about in terms of artificially manipulating memories, because the theme here will be that artificially manipulating memories can be not just a very scientifically important endeavor, but I think one that can also have therapeutic purposes, and one that really, again, imbues memory with more power than perhaps we've appreciated before. So, okay, to start off here, imagine that you have a rodent that's going around and exploring what we'll call environment B. The rodent is running around, it's forming a memory of this particular environment, and then we give it a couple of mild foot shocks, right? It doesn't hurt, it's more like a static shock, if anything. There's a surprise factor here. And we give the animals a couple of mild foot shocks to form a particular negative memory, I'll call it, of this environment. And now at the same time, while this is happening, with the genetic system that we're using, we can find the brain cells that are holding on to this negative experience of environment B. And what we've done is we've tricked those brain cells to respond to brief pulses of light, so that you can go in and actually shoot lasers. You can optically go in and shoot light into the brain, into the patch of hippocampus that has these cells that are holding on to this negative memory, so as to photoactivate them. The brain's a pretty dark place, so it doesn't naturally respond to light other than your retina. So this is an artificial means to get some kind of optical control over cells in the brain that hold on to a memory. So again, the animals here are given a couple of mild foot shocks and they form a negative memory of environment B. We find the brain cells that hold on to that particular memory, and we trick just those brain cells to respond to light. Now, the million-dollar experiment here is, if we put the animals the next day in environment A, this is a completely different environment with completely different sights and sounds and smells associated with it. The animal has no reason to be fearful of this particular environment. The million-dollar experiment here is what happens when we shoot light into the brain. And then while they're in environment A, we activate the memory here that was formed in environment B. Will the animals behave as though they're in environment B? In other words, would they recall the negative memory? And the way we measure this in animals is also a conserved behavior called freezing. When an animal, or when a rodent in this case, is fearful of a particular environment, one of the many behaviors it'll display is what we call freezing behavior. And it's so-called freezing behavior because the animal freezes in place. It stays in place, so presumably as to not be detected by a potential threat. So freezing in this case is complete immobility. You could think of it as like if a really loud noise were to happen right now, your first response might be to tense up and then to look around to see what happened. You're freezing in place, and then fight or flight and a myriad of other behaviors kick in. So that's what we're measuring here is when we activate these brain cells that hold on to this negative memory of environment B, do the animals freeze even though they're in environment A? So this is the video of what we saw almost a decade ago now, and again, this will be the launching pad for the rest of the talk. You can ignore the caption. The first half of this video is just a control. In other words, we're not activating a memory. We're just shooting light into the brain to control for the effects of light. This is as expected here. The animal is still running around, it's sniffing around the corners. It's not displaying any freezing behavior, which is great. This is the same animal. It does have the negative memory of environment B, now photoactivatable in its hippocampus. And when we activate those cells, you can see here that the animal almost immediately goes into this freezing posture. So said another way, going in and optically activating cells that harbor that memory of environment B, that negative memory, was sufficient to drive freezing behavior. And this was really exciting for us because it gave us optical leverage over a given memory. It was a proof of principle for us that we could go in and start tinkering with the stuff of memory in rodent brains. And as I'll talk about in a bit, it really opened up the floodgates for studying different kinds of memories. We studied negative memories in rodents here, given their applications to studying rodent models of PTSD and anxiety, which I'll also get into, but also here because the behaviors that we were reading out were behaviors that we could go in and quantify. Like we could put a ruler to the animal's behavior and ask, was it freezing when it shouldn't? In other words, was it recalling this negative memory or not? And I will say this, there was a lot of bag of tricks here that have really come up in the last decade in terms of what kinds of memories we can do and in terms of what it means to activate brain cells that hold on to a particular negative memory. So to give one example of the curveballs that nature has really thrown our way, I'll briefly go over a quick project from Caitlin Dorst, who's a phenomenal grad student in the lab right now, who is really asking this question of, okay, we can go in and we can artificially activate negative memories, but what does that mean, right? Like there's some days where you might recall a negative memory and it might motivate you to do one thing, like to go to the gym or go get food. Another day, it might motivate you to continue to just stay in bed or stay on the couch or all things in between. So there's a whole host of behaviors here that a given memory can promote, but we were asking, can these cells in the hippocampus that are holding on to this negative memory, when we artificially activate those cells, would that always produce the same behavior in the animals? Or was it flexible? Could these memories drive different behaviors depending on what the animals were undergoing? So for example, let's say we have the animals that are given the fear memory of environment B, the exact same as before. The cells that are involved in holding on to this fear memory are now photoactivatable. And then we put the animals in a handful of different environments. There's a small box, a medium box, and a large box. We wanted to keep it as simple and as straightforward as possible. The small box, it's almost like the size of a shoe box. The animal doesn't really have as much room to roam around. The large box, you could imagine it's like one of the boxes you use when moving out of a house, for example. So would activating the same negative memory across these different environments produce the same behavior? Like once a negative memory, it always manifests as freezing, for example, or would it be different depending on the environment that the animal was in? Now, I'll walk through this really quickly here, so bear with me with the data, but what we're seeing here on the x-axis is whether or not the cells are turned off or turned on by light. And then on the y-axis, we're looking at freezing. So in other words, the amount, quote unquote, of fear that the animal is recalling. And now the most important point here is between this red point and the blue point. When we turn on these negative memory cells, the animals do show freezing behavior. They start behaving exactly like in the video that I showed you before. This is something that we've known, that these cells will produce, these cells are sufficient to drive that negative memory. But what we found surprising was that when we activate those same exact cells in the large box, in this case, the animals don't freeze, like at all, actually. If anything, they're perhaps maybe even promoting some more escape-related behaviors. Like Caitlin uses an example in her talks of, if you see a bear in the woods versus a bear in a large open field, whether you decide to actually run or to hide and so on, your environment dictates how that experience actually ends up manifesting as a behavior. So this was really interesting for us because it meant that these cells in the hippocampus that drove negative memories were in fact flexible in the kind of behavior that they produced. So a given negative memory and the cells holding on to those can produce different behaviors. It doesn't always have to manifest as the same way. And one of the goals of this project now is to figure out why and which brain areas are mediating this. But okay, so the first third here of the talk is looking at this idea of artificially manipulating negative memories. And for the next third, I'll go in and talk about what does it mean to even manipulate a memory and what happens to that memory. And then I'll end with applications of how memory manipulation can be used in a therapeutic-like manner. So when I think of memories, I think of them as the thing that our brain does to thread and unify our overall sense of being, right? It threads our identity over time. It's our conscious self propagated over time, and memory happens to be this sort of magical bow that ties it together. Now, one of the bitter ironies here is that one thing that we've really learned in the past like half a century or more of memory research is that memory itself is not like an iPhone video of the past, but it's more like, imagine that you're putting up a photo on Instagram and you're constantly applying filters to it, for example. The filtered picture in the end is going to be kind of different, but kind of similar to the original picture. And we think of memory as the same way, where when we recall a memory, it becomes unstable. Like memories are constantly being modified and updated with new information. And then we say that that process is memory being malleable. So that's one thing that we really wanted to now lean into, is this idea that memories are warpable, they're malleable, and that's good and bad. It's good because it means that our past isn't, our conscious access to the past is not set in stone. It can be bad in the sense that memories can become unreliable, with false memories being an extreme example of that, to say nothing of eyewitness testimony, of course, in the court of law. So one of the projects that we took on here was this idea of, can we leverage memory's malleability and then try to alter or warp the contents of a given memory? So first is the proof of principle, can we do that? And then the follow-up to that would be, is there any therapeutic setting where we could actually utilize that in a beneficial manner? So this is something that is pretty near and dear to me because I've been fascinated with false memories for over a decade now, and our culture has too, right? For anybody interested in the idea of false memories, it permeates our culture left and right. I mean, Hollywood and cinema and like TV are kind of obsessed with the ideas of memories because of how deeply connected and intertwined with our sense of self they actually are. Black Mirror and the newest Blade Runner being a great example of this for anyone interested. And just to really drive the point home of the idea of warping memories, or how easily memories can be warped, there is one experiment here that I'd like to do with everyone in the audience that is a testament to memory's malleability. And it's also a great way to have like an existential crisis at night, thinking about, well, if memories are really malleable and they're basically warped every time we recall them, yet memory is the thing that threads and unifies our overall sense of being, what does that say about our overall sense of being? That it might be a little bit more unstable than we originally expected it to be. So for instance, one of the well-documented cases here of false memories permeating our day-to-day routines is the so-called Mandela effect. So for those interested, look up on BuzzFeed this 20 examples that will make you believe you're in a parallel universe. And it basically all too easily points out how our recollection of the past can be easily warped, or I should say, how much of a misrepresentation of the past we may actually have. So for example, for anybody in the audience, right, there's two pictures of the Monopoly Man here. Is the Monopoly Man the one with the monocle or without the monocle? The other example would be Queen's song, We Are the Champions. Most of us remember the final lines of the song being, we are the champions, we are the champions, we are the champions of the world. And then everyone starts yelling and Fenway Park goes crazy and it's a great time. Now, in these particular examples, it turns out that the answer to which one is really the Monopoly Man is the one on the right, and about 50% of people actually misremember that it's the one with the monocle. What I think is happening is that the Planters peanut guy has a monocle, The monopoly man does not. And I think that somewhere in our brain, at some point, we just warp the two together. And that's just the way that half of us tend to remember this. The other part is with Queen's song, We Are the Champions, the line of the world is never said as the last line of the song. The song just ends with We Are the Champions, and that's it. And then yet it's also like about half of people tend to misremember that the last line of the Queen song is of the world, even though that isn't necessarily the case. One of my favorite recent ones, because I still get this one wrong all the time, and I happen to be a big peanut butter aficionado. So this is with Jif, Jiffy, and Jif, right? Which one is the peanut butter one? Give you a second to think. And the answer is that it's this one. And I think that what ends up happening here is that some people that misremember Jiffy is one of the more common answers, probably combined Skippy peanut butter with Jif to give you Jiffy. And then finally, probably the most famous one of the bunch here, in the movie Star Wars, Darth Vader never says, "Luke, I am your father." That's probably like the most misquoted line in cinema. It's subtle, but he actually says, "No, I am your father." And that small subtlety though is pretty facetious here in the scale of cinema. But imagine that being the reason why someone is actually thrown in jail or not, right? You can see the ramifications of misremembering here all too clearly. So okay, what we wanted to do to try to create a false memory in the lab, and then I'll end again with applications of why this is important. One way of going about this was by using the same strategy that we talked about before with finding brain cells in the hippocampus that hold on to particular memories and to trick them to respond to light. So the behavior is just slightly different here. So we'll take our rodents, for example, and we'll put them in environment A, right? It has its own unique set of sights and sounds and smells associated with this particular environment. While the animals are forming a memory of this box of environment A, the cells that are holding on to the memory of box A are now being tricked to respond to pulses of light so that we can both visualize them under the microscope, like the first images that I showed, and so that we can optically control them. So, okay, we have optical leverage now over the memory of environment A. The next day, the animals are put in environment B, and this is where they're given the couple of mild foot shocks to form the negative memory of environment B. But the key intervention here is that while the animals are forming the memory of environment B, we simultaneously shoot light into the brain to activate the memory of environment A. So it's an attempt to artificially connect the two, the memory of environment A with the mild foot shocks that are actually happening to the animal in environment B. Because if that works, then what may happen is that when the animals are back in environment A, where technically speaking nothing negative happened, they should behave as though this is where they were given the mild foot shocks. In other words, they would freeze in A, even though the negative experience itself took place in environment B. And this is what it looks like. This is the animals exploring environment A. They're running around. The cells that are involved in harboring this memory of environment A are being tricked to respond to pulses of light in this case. Now, the animals here are given a couple of mild foot shocks while we're optically reactivating the memory of environment A. You can see that here environment B is very different, and then presumably the animal should quote unquote be thinking about environment A. And now when we place them back in environment A, you can see that they almost immediately go into this freezing posture. So they stay still here in place. Now, this was also exciting for us because it really gave us a proof of principle that we could leverage memory's malleability to thereby create a kind of false memory in the lab. And we were excited by this because it's not so much the fun and games of creating false memories, although I'm a humongous sci-fi Inception fan and so on. It's more because if memories are as malleable as they appear to be, then we can imagine certain instances of people living with PTSD or people living with generalized anxiety, for instance, where we could try to actually leverage this malleability inherent to memory in a therapeutic manner. I look at this as like a cup is half full approach here, where memories being malleable can actually be, I think, in my opinion, a very beneficial thing because we can utilize that malleability to try to mitigate suffering in any capacity, especially in as much as negative memories are involved in producing some of that suffering. So that's what led me, and finally here to the application part of this, with regards to, okay, we were able to artificially activate negative memories, we're able to create false memories. Now, two different things. Could we do this with positive memories? Positive memories almost feel like they inherently have some more therapeutic value associated with them. But then also, could we activate positive memories to begin with, and then do so in animal models of PTSD and depression and anxiety in particular? Now, with regards to positive memories, this one got interesting because we know that for humans, right, we all have our own version of positive experiences. We all, if you think really deeply, you could think of some of the most positive experiences that you've ever had, and they change. They alter your physiology. They change your thought process. They change your behavior. So they're pretty potent. And now I use this example of positive memories not so much because I was born and raised in Boston and I'm a big Boston sports fan, but because just to highlight how powerful positive memories really can be. So for instance, two of my favorite positive memories of literally of all time are either Malcolm Butler's interception in the Super Bowl that won the Patriots the Super Bowl, or the Julian Edelman catch when they had the big comeback against the Falcons in the Super Bowl. Now, outside of it being kind of like a groany joke of these kinds of like Boston-based sports memories that I have that are near and dear, these actually did serve a purpose that it took me really thinking deeply about this to realize how important these kinds of positive memories were to me. So for instance, I used to have like serious anxiety about public speaking. I used to hate it more than anything else. I mean, like borderline panic attacks before speaking in public. It was basically my kryptonite, and it was just something that I didn't really know what to do about it outside of practicing and just continuing to give talks. It was pretty crippling, and I took classes. I did everything I could to try to really overcome this fear of public speaking. And what actually ended up helping more than anything, besides maybe just the art of practice itself, was what I would do before giving a talk or something that I was super nervous about talks being up there. What I would do is I would watch some of these clips of Patriots highlights of them winning Super Bowls, and it would put me in such a good mood, and it would put me in like a my adrenaline matches my heart rate, matches my sweaty palms, like my physiology just kind of makes sense now. As opposed to why am I super anxious before a talk and my heart rate is off the charts and so on. So I'd use these experiences as a way to almost like hype myself up, or to really to think positively and talk positively to myself. And they would put me in this kind of like adrenaline filled, okay, I'm ready to conquer this talk, and I'm ready to go into the room and speak in front of people. And it really worked. Like this was something that it was really like a potent kind of memory linked intervention here that I would use on myself before doing something that made me supremely nervous, until over time until it didn't make me nervous anymore. And now I can just watch these and enjoy them just for the pure hedonism that Boston sports is. But yeah, so positive memories are powerful. And there is thankfully a burgeoning body of literature that's emerging that asks two questions. The first is, what can we do to think outside the box in terms of intervening with the brain in general, especially when it comes to any pathological states? And then the second is, can positive memories be viewed as something that's now in our arsenal of a toolkit to go and manipulate the brain, for instance, in a therapeutic manner? We have a slew of drugs, of course, that can do this, but can we actually go and give this kind of interventional power to positive memories in particular? So for instance, one commentary that I really enjoyed reading because of how much of a sobering reminder it was from former NIMH director Steve Hyman, who wrote this commentary in Science about how, despite the plethora of different drugs that are available for treating disorders of the brain or mental health disorders, the efficacy of these treatments is pretty strikingly low, nowhere near as good as we would hope, of course, let alone something coinciding with a cure. And the call to arms here in this commentary was that it seems like we got lucky in the 1940s and 50s when it came to discovering some of the targets in the brain that modern antidepressants and anxiolytics target, for example. But we kind of stopped thinking outside of the box. And what I mean by that is, we need the drug work, of course. It's the most non-invasive, one of the most non-invasive ways we have of being able to try to treat the brain. We need the drug-based research more than ever. We also need to think outside the box and ask, are there any non-traditional drugs or therapeutics that we can use to really try to alleviate the brain? And then my argument here is that positive memories may be one of them. So this was basically a call to arms that this neuropsychopharmacological revolution that we had really witnessed in the 1950s and 60s kind of stalled. And then how do we jumpstart this revolution again? I like to think of this as like, you know, if we use today the same telecommunications devices in 2022 that we used back in 1949, everybody would say, well, telecommunications has just simply not progressed in the last 70 years, right? But we have progressed to like iPhone gazillions now, and like basically all these supercomputers in our back pockets. Why hasn't the research when it comes to treating the brain undergone a similar revolution that our phones have, right? There's many answers to that. And I think that this is just more of a kind of fanning the flames here of being this call to arms of continuing to think outside the box. Now, there's been, thankfully, many groups that have really taken up this idea of the capacity for positive memories to intervene with our behavior. So with our cognition and behavior. So for example, there's a fantastic study a couple of years ago about how simply thinking about positive memories helps to really bring down the body's stress responses. This includes everything from heart rate to blood stress hormone levels and the works. These recalled positive memories also activate a lot of the brain's reward circuitry. So that's exciting because it means that there's a potential answer already in the brain for how we can intervene with the brain itself in a beneficial manner. Now, a follow-up study here basically confirmed this idea of this power of positive memories, was that even thinking about negative experiences and then trying to extract some kind of positive meaning in them, this is maybe kind of the basis of mindfulness too, but if we can go and extract some kind of positive meaning out of a negative experience, it's both adaptive, but also it takes advantage of memory's malleability in a positive manner, because we can, as the title suggests, finding these positive meanings in negative memories can actually mitigate some of that negativity in that memory, which is something that I'll directly talk about in a few slides. Now, this built off of previous work from 7 years ago now, where we started with a basic hypothesis. Positive memories, when artificially activated, in this case in rodents, are enough to alleviate pathologies of the brain. That was the straightforward hypothesis that we had when beginning these experiments. Now, with rodents, there's all sorts of positive experiences, right? In humans, it's easy, right? Your favorite food dish, maybe socializing with your best friend, a great phone call, your favorite Netflix TV show, everything in between. In rodents, they have a similar buffet of positive experiences. It could be socializing with each other, for instance, certain food rewards and certain sugary, watery rewards, kind of like soda for people that we also enjoy. So we use these experiences to give the animals positive experiences. And once again, we find the brain cells that hold on to these positive experiences and trick them to respond to light. The first finding that really sets the stage for what my lab is currently doing in a handful of projects was that we're able to produce animal models of stress, which precipitates various behaviors that we think are consistent with depression and anxiety. And the short of it is that artificially activating positive memories was sufficient to reverse these depression and a subset of anxiety-related behaviors. That was probably one of the findings of a lifetime for me personally, because having been somebody that's either 0 degree and or 1 degree removed from these mental health disorders, it was really meaningful to engage in projects that could actually pinpoint to novel solutions and continue to think outside the box. So positive memories here were sufficient to reverse behaviors associated with depression and anxiety. And now we wanted to ask, in this case, could we use positive memories as an intervention for certain animal models of PTSD to start off with? So for example, this was a project led by a former postdoc in the lab, Stephanie, who asked this question of, can we go in and activate positive memories while an animal is recalling a negative experience, and to see what happens. So we'll start again. We go in and we give animals a couple of mild foot shocks to produce this negative experience. The next day, the animals are placed back in the same environment so that they're recalling the same negative experience, but at the exact same time that they're recalling the negative experience, we artificially activate the positive memory. In this case, it was them socializing with some of their cage-mate buddies. So the animals are recalling the negative experience in environment B, but while we're simultaneously activating the positive memory from a few days before. And now what we see here is pretty dramatic. So first we see here, we have here on the x-axis whether memories are reactivated or not. On the y-axis, we have the readout of fear that we're using. In dark red is the group that had the positive memory reactivated. And then in pink is just a control group. So first, when there's no positive memory reactivated, both groups, the positive memory group and the control group, have about the same level of fear. So they recall the fear memory about the same extent. Now, what was really exciting to us was that when we optically artificially activate the positive memory, while the animal is recalling the fearful negative memory, they show a dramatic decrease in fear responses in real time. So in real time, we're going in and activating a positive memory, which is sufficient to suppress the negative behavioral responses associated with fear, whereas this doesn't happen in the control group. And to summarize this study, that it should be, everyone can mark their calendars. I believe it's coming out September 12th, or the week of September 12th, it'll be coming out. But the short of it was that not only does artificially activating a positive memory suppress these negative fear responses, we actually developed a strategy to suppress them permanently so that we can activate a positive memory, take advantage of how malleable memories actually are, and in a sense, rewarp that negative memory to have less negativity associated with it. And so activating a positive memory, we were able to successfully do so in a manner that permanently suppressed those fear responses. So said another way, artificially activating a positive memory in an animal model of PTSD permanently interfered with that negative memory. It permanently rewrote it. We have good evidence to believe that it actually rewired what that memory, how that memory was represented in the brain to begin with. So at least one of the examples here of being able to utilize artificially activated memories, ideally in a manner that suppresses symptoms associated with PTSD, for instance. Now, okay, in the last leg of the talk here, I want to really lean into this idea that that memories are what we say distributed throughout the brain. So when we think of a memory, the sights and sounds and smells and emotions and environment where that memory was taking place, those different qualities of memories recruit different parts of the brain that are involved in processing the sights and sounds and smells and emotional components of that given memory. That's a long-winded way of saying that memories don't exist in a single XYZ coordinate point in the brain. They're distributed. They recruit the brain in 3 dimensions. Like memories recruit the brain high and low, left and right, ancient and modern. And here we're asking, rather than going in and looking at a cross-section of the brain, as I showed you before with some of the images of what the cells holding on to memories look like, could we do so in a 3-dimensional manner? Could we actually go and visualize memories in 3D in the brain? What would that tell us about memory? And ideally, can we get greedy with this, and instead of visualizing 1 memory, could we visualize 2 memories? Like let's start there, right? Two is more than 1, and we can go, and if we can visualize 2 memories in the brain, we can ask, how do they interact? If it's a positive and negative memory, for instance, in particular. So to do this, Monica Spicchetti, who's a PhD now, former grad student in the lab, took on this idea of asking how are positive and negative experiences, memories, manifested in the brain, and can we develop tools to track both experiences in the brain of the same animal? All of our brains hold millions, or countless, I should say, numbers of memories. So we wanted to start with, we've previously been able to artificially image 1 and activate 1. Can we do so with 2 in the same brain? And hopefully within our lifetimes, 1 to 2 turns to many in terms of how we can do this. So to do this, she really spearheaded a handful of genetic tools in combination that were able to make it so that we could find the cells that held on to a negative experience, which is visualized here in red, for instance, and we could find cells that hold on to a positive experience, which are visualized here in green. Now, the first remarkable finding of this paper, which is in press and should also be out in about a month, the first remarkable finding, in my opinion, was that the neural correlates of negative memories in an area of the hippocampus that we're looking at here, and the neural correlates of a positive memory, same area of the hippocampus here, seem to be largely segregated. In other words, there seems to be some kind of pre-existing landscape in the brain where negative memories are likely to land and where positive memories are likely to land. That was exciting for us because it meant that, even before forming a memory, we might be able to pinpoint where a positive experience is going to go and reside in the brain compared to where a negative memory might go and reside in the brain. Now, we find that useful because it means that there's two, to put it in a fancy way, there's two topologically distinct features here of positive and negative memories. And if we can go and actually find the different landscapes of each, that by definition gives us different targets, right? This goes back to thinking outside the box now for interventions of the brain. If we can intervene with areas of the brain that are involved in positive memories, we could try to do, for example, what we did in rodents with intervening with symptoms associated with PTSD. Or if we can do so with negative memories, could we go in and try to turn negative memories off, for example, and see what that may tell us about the brain? So the first thing here was that positive and negative experiences are processed in distinct set of cells in this area of the hippocampus here. So the second part here, though, was asking like, okay, that's cool. That gives us this key physical difference between the two, but what can we do with that data? So what we did with that data was we asked, could we go and take these brain cells that hold on to a positive experience and actually go in and sequence their genome? Can we go in and actually sequence the genetic landscape of these cells and ask, is there something that's different about the genetic landscape of cells that hold on to positive memories compared to cells that hold on to negative memories? You don't have to worry about the numbers here. It's basically saying that there was a lot of genes that are upregulated in positive cells. There's a lot of genes upregulated in negative memory cells and some in both. And now these two tables here are, you don't have to go in and like read them all, of course, but the two tables are just to drive the point home that there were a lot of genes that were different between cells that held on to positive memories and between cells that held on to negative memories. So even at the genetic level, the landscape of positive and negative memories seem to be distinct, that there seem to be distinct genetic signatures for positive memories that are very different than negative memories. And in an almost twist of irony, and this is something that we're really following up on heavily, and I'd love to be able to basically invest the next 10 years of the lab in these styles of experiments. One of the things that we found in this data set was that the cells that were involved in holding on to positive memories were enriched in genes that we typically think are associated with protecting brain cells. So they were involved, these genes were involved in metabolic upkeep and some upkeep processes to prevent cells from dying, for example, and we call this neuroprotective in this instance. That positive memory cells seem to be enriched in genes that were involved in protecting brain cells. And now the flip side of that was that the cells that held on to negative memories were in particular enriched with genes that have been traditionally associated with Alzheimer's disease. So this was a big surprise to us because these negative memory cells were enriched in APP and ApoE, and again, some of these canonical gene markers that we use to study Alzheimer's disease. And then that really got us thinking that the same way that there's a burgeoning body of literature suggesting the power of positive memories in alleviating aspects of our cognition and behavior, negative memories or negative emotions and experiences are at the core of a plethora of psychiatric disorders and neurodegenerative disorders as well. So could there be some kind of causal link here between cells that process negative memories and Alzheimer's, for example? Huge line of research that the lab is really excited to dive into. And then likewise, could there be some kind of prophylactic capacity or protective capacity for positive memories to provide a kind of protective net over the brain to even prevent the brain from degenerating to begin with, right? In this case, in other words, would negative memories be sufficient to cause degeneration in Alzheimer's in the brain? And could positive memories be used to either restore the brain back to a healthy state or to even prevent the brain from degenerating in particular? So we're knee deep in these styles of experiments now to see where it's going. And the short of it is without, I guess like this is kind of half spoiler, half not spoiler, because we're still doing these experiments. But the short of it is that if you repeatedly reactivate negative memories in the brain, it produces a battery of cognitive and behavioral deficits. Now, if we activate positive memories, this actually seems to be enough to either reverse certain pathologies of the brain associated with psychiatric disorders in particular. And we're asking now we're asking, can we artificially activate positive memories to prevent the runaway train that neurodegeneration is? And then hopefully in due time, I'll be able to update on the state of all of those projects. So to end here, I want to end with an example of why this matters and what are we doing. And like I skipped one slide because I want to leave plenty of time for a Q&A here as well. But the previous slide was essentially being able to artificially control negative memories to prevent the return of fear, consistent with the theme of the talk. But okay, so we're going in and we're shooting lasers into the brain and we're artificially activating memories to kind of force memories to be recalled in rodents. Why? And what does that tell us? I think the why is from the basic science perspective that knowing is better than not knowing, and that knowing more about the brain and how the brain works will make us better equipped to figure out what happens when the brain breaks down. Now, the what have we learned is that the idea of memory manipulation is not just here and with us to stay, but something that I think can be used both responsibly and in a manner that alleviates, again, symptoms associated with psychiatric and neurodegenerative disorders as well. And that's exciting because that's taking up the call to arms of really thinking outside of the box on what can we do to try to prevent disorders of the brain or to try to restore the brain out of disorder and back to a healthy state, for instance. And I think that, I mentioned with the example of artificial memory manipulation and how we've shown here that we can jumpstart that process and use it to alleviate certain symptoms in rodent models, for instance. And I hope that it can have the kind of therapeutic or prophylactic or both, like clinically relevant application that drug-based work does today as well, right? Both are going in and intervening with the brain. Like both are going in and tinkering with the stuff of thought and asking, how can we use this in a manner that like lets us know more about what's happening in the brain, or ideally that also lets us go in and intervene when the brain enters a disordered state, for example. So I began with, what does memory mean to you? And hopefully throughout this talk, the answer to that question has changed a little bit. Like what memory means to you, I hope is something that maybe is a little bit more enriched. Like it's a process that happens in the most complicated organ that we know in our universe, right? And yet you do it without even thinking. Like that's how seamless and that's how smooth out this process actually is. Not without its hiccups, as we've seen. So in Inside Out, right, it's like there's examples of memories that are sad here in blue, or memories that are a bit happier here in gold. Showed here basically the science version of this is being able to go into the brain and visualize cells that hold on to discrete memories and tinker with them. And ideally to really leverage this idea that memories actually exist in complicated fashion, right? It's not just that a given memory is sad or that a given memory is good. They are imbued and enriched with the kind of information that makes certain memories worth recalling and makes other memories seemingly worth avoiding as well. So I do kind of want to tip my cap to the phenomenal process that memory is, and hope that the answer to the question of what does memory mean to you is not just a bit more rich, but a bit more nuanced too, that this amazing faculty that we have to be able to recall virtually any experience of our past happens both so seamlessly, can change our mood and behavior and how we conduct ourselves and what state we're in the works, and that ideally we might even be able to use that process in a manner, again, that happens to restore processes of cognition and behavior, or that happens to alleviate the brain as it enters a pathological state as well. So, okay, with that said, two things. One, humongous thank you for your attention and for showing up today like this. It really means a lot to see so many people here. And I do want to give the biggest shout out of all here to the lab. It's like the lab they're the ones that were in the trenches doing these experiments and really going to town on this idea of memory manipulation, where it's going, and then discovering more and more about the uncharted landscape that memory is in the brain. for those interested, we have a lab Instagram account that goes and shows basically what the day-to-day grind is like in the lab, as well as some celebrations as well. My Twitter account, where I try to be a force, an anchoring force of positivity. Like you may have noticed from my tone, I'm very much a cup is overflowing kind of person. So there's an inherent optimism that I like to embody. This is just like my parents speaking through me right now, but there's like an inherent optimism to how we approach things, whether it's our day-to-day grind or whether it's trying to alleviate pathologies of the brain as well, and seeing what that tells us. Like I know that we can do it, right? I've decided to dedicate my life to it because I believe in it, and I'm really thankful that everyone in lab believes in this process that is science as well, and to really leverage what memory actually means to us. So. First and foremost as well, I mean, thank you to the lab and everybody here. Thank you to everyone for your attention. To all our funding sources, without this, our science simply can't be done. We're really excited that you decided to take a chance on this idea of artificially tinkering with memories, which hopefully has, in the long run, yielded some good in the world. So thank you very much for your attention. I'm very happy to take questions.
Good. Steve, thank you so much for your presentation and, more importantly, the extraordinary work that you're doing. We see the wonderful team of obviously brilliant young scientists that are working with you. And I think your positivity just reverberates in your presentation and is shared with all of us. So thank you for all that you're doing. I want to ask a little bit as you think towards the future of this. We know that interventions that certainly are other than medicine can affect the brain, such as cognitive behavioral therapy. We know that exercise has a positive effect on the brain. I'd like you to speak a little bit more about how people can use their positive memories to help them feel better, to help them cope with difficult circumstances, similar to the examples that you gave in your own life. What in the here and now can people do?
Yeah, absolutely. I mean, this is a fantastic, fantastic question. And I think that the here and now, to put it really concretely, is let's take, for example, journaling and having a gratitude journal. Gratitude journal, you can get some for like $10 off of Amazon. And essentially, the one that I use, for example, it gives you 3 small prompts. I do it in the morning. It gives you 3 small prompts to really sit with. Like, think about something that happened yesterday that made you happy, or think about a time with your parents or sibling or brother. Like, it gives you pretty specific prompts to write one sentence in this case. So it's super straightforward to write one sentence to answer the prompt and then to sit with it a little bit. This has been something that time and time again, psychology studies have shown us that gratitude journaling is one of the many ways to increase like our both mood and positive outlook on life, but also again being able to do what one of the studies that I showed, to be able to extract positivity out of an otherwise pretty chaotic world, to put it mildly, right? Like everyone has every good reason to be on edge and stressed these days, and it's like kind of reached fever pitch here, understandably. So taking those moments to pause, and something like gratitude journaling is a very direct example, but it can be more like practicing mindfulness or being comfortable. Sitting with your own thoughts is a really hard thing to do, but I think like a workout, you can get better and better at it, and you can get more and more efficient with that process. So one thing that I like to do with gratitude journaling in the morning is that one thing that I've noticed in me, at least in terms of palpable changes, is that it not just helps me keep things in perspective, but it helps me know that despite what I'm seeing on the news, or despite having maybe a really stressful day at work, or a lot of hard things that are happening in my life or in others' lives around, that I still feel empowered to do something about it, and that what I'm doing matters, and ultimately that my experience in the world matters and is valued. And I think that really hits at like a pretty deep-seated human yearning for purpose and meaning, for that matter, and being able to find that purpose and meaning in our memory banks is such a powerful thing that again, like time and time again, psychology studies are suggesting help improve our overall well-being. So that's one example that comes to mind that I can definitely vouch for.
That's a great example, and I think a part of what the science does, the work that you and your team have done, shows that this is there's a physical aspect to when somebody does this, that these kind of activities really can affect the brain physically. And I think that often is helpful to people to understand that the actions that they take really can affect their brain in a positive way and then affect their mood and how they manage challenges and difficult circumstances.
Exactly.
We have time for one more question. I'm going to ask you, in 10 years from now, where do you see that this work will bring us to?
Oh yeah, in 10 years from now, I hope that, like with the Human Genome Project, where we started off being able to sequence a few genes, and then 20 years later, the whole thing was sequenced. Ideally, if we could have these kinds of three-dimensional maps of what positive and negative memories look like in the same brain, that'll be a start and my hope for the next 5 years. But then in 10 years, can we look at how these cellular substrates for memory, how do they change as the brain deteriorates, for example, in particular into Alzheimer's and neurodegeneration in general. So if we can have a map, like a dynamic map of this is a physical basis for a positive and negative memory in the brain. Here's what it looks like at time 0. Here's what it looks like when it's entering the degenerated state. And here's what it looks like when it's in a severely degenerated state. I'm hoping that those kinds of maps can also provide a roadmap for what potential treatments might look like. Which cells are deteriorating faster and why? Is there a certain corner of the brain that are hit harder than others? Certain kinds of memories that are hit harder than others, and so on. So I think that that would be a bit of a dream for the next decade, would be to have these three-dimensional maps of multiple memories in the same brain, and then to be able to track how those memories change as they enter the pathological states that exist in the brain as well.
Well, I'm looking forward to, before a decade from now, before even 5 years from now, having the opportunity to get an update from you. And once again, thank you for a wonderful presentation and the extraordinary work that you're doing and your team is doing. Thank you so much.
Thank you too. It truly has been a pleasure, Jeff. Thank you very much.
Thanks. I also want to thank everybody for joining us today. And I'd ask you to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We can do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientists. To make a gift, please visit bbrfoundation.org or call 1-800-829-8289. This webinar has been recorded. If you've missed any portion of this presentation or would like to share it with family and friends, please visit the events and webinars page on our website. And again, I hope you'll join us next month on September 13th at 2 p.m. Eastern Time, when Dr. Brian Brennan, Medical Director of the Obsessive-Compulsive Disorder Institute and Associate Director of Translational Neuroscience Research at McLean Hospital and Assistant Professor of Psychiatry at Harvard Medical School, will present identification of symptom-specific brain circuit abnormalities in obsessive-compulsive disorder. Once again, thank you for joining us, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you very much.